Hello. Thank you. Appreciate it. No, it is a pretty good morning if you don't have allergies. Does anybody have any questions, concerns, clarifications? Anything at all? Concerns you want to share? So the question is, have I decided how I'm going to curve the second test? The answer is, as I've been telling some of you in office hours, it makes no sense for me to tell you how I'm curving each individual test because really the curving happens at the end of the semester. So no, each individual test, I won't be assigning you a letter grade based on a curve for that exam. You'll find out at the end of the semester. In fact, right before the final, I will probably try to give the class some sort of an overview projection of what's happening going into the final, but until then, it's really hard to say. Other questions? Topics? Things you're confused about? Things that we've missed? Yeah? So I said the magic word. Yeah.
Well, we're stepping into topics.
Yep.
So I'm comfortable with the gene mapping.
Okay.
I'm trying to figure out how that relates to p-value.
Yes. So the correlation or how genetic mapping is related to statistics. That's why I included at the last minute, which I'm glad I thought of now, especially since you said that, an example today of how genetics, and we'll carry that into next class, how genetics and gene mapping uses chi-square testing. So thank you very much. You're exactly right. That's where we're trying to head. Done chi-square tests. We've done a lot of examples now of genetic mapping. Finish those today, and then we'll move on into some other minor topics, complex traits and genetic hitchhiking. You've seen this question come up the last couple of classes. This is the last time you're going to see it. I just wanted to explicitly point out that the information I'm going to give you, if, for example, I want you to do something like do genetic mapping between the genes B and L, which is generally what's always going to happen when you see this sort of a cross, dihybrid test cross. It's almost always going to be asking you what's the genetic distance, centimorgans, map units, recombination fraction between those genes. I'm going to give you this information. I'm going to give you numbers of observed F2s, and you're going to have to figure out, deduce, what are their genotypes, and then figure out from there, are they parental or recombinant type genotypes, and from there calculate genetic distance. So from here, if I've already given you the recombinant numbers, this is again just review. Recombination fraction is going to be the total number of recombinants, 67 plus 58, the R types, over the total, which in this case happens to be what? Yeah, so the total's 500 observations. Total numbers of F2s we looked at. And 67 plus 58 is 125. Into 500 is 25 centimorgans. So that's the answer to that question. The genetic distance between B and L in this case is 25 centimorgans. How would you draw a genetic map that represents these genes? I said, draw me a genetic map. Now that you have the distance, what does a genetic map look like? What sort of information do we now know about these genes? Okay, aside from the fact that they're 25 centimorgans, how do you interpret that?
You're going to draw a chromosome.
So here's a chromosome.
And you're going to put B and L on either side. You're going to connect them by the 25 centimeters.
So how did you know that they were on the same chromosome, as opposed to being on different chromosomes?
Less than 50.
Because the distance of 25 centimorgans is less than 50. So what do we know, review again, about the magic number of 50 centimorgans? They're either going to be, right, so 50 centimorgans either looks like this, or like this. B is on one chromosome and L is on a different chromosome. Or they're at the opposite ends, extreme opposite ends of one chromosome. So if it's anything less than 50, we draw something that maybe looks like what's up in the upper right here. B and L, they're 25 centimorgans apart. So there's a genetic map. We know from this information that B and L are on the same chromosome. Any questions? Please.
Will we also be given a parental types, or will we just make those the same?
So you have to be able to, right, there always has to be a number of pieces of information. You have to know the P zeros because you have to know which type is recombinant, which type is parental. So how did I do that for this example? How did I put these RP designations, parental type or recombinant type? How did I get those? So it's which, you compare the F1's gametes to the P0 gametes. And this is the same thing we've seen for a few classes now, but I'm just saying it in new words. So the way that a geneticist detects recombination is by comparing two successive generations worth of gametes, the P0 gametes and the F1 gametes. You want to look at the material, in other words, that went into making an organism, the F1, and then you want to look at the genetic information that comes out of that organism, the F1. So you want to look at the sperm and the egg that made the F1 individual. And then it's inside the F1 individual, when they're doing gametogenesis, when meiosis is happening in that F1 individual, that's where crossing over is happening. So it's the F1's haplotypes, their gametes, that are going to demonstrate evidence of whether or not recombination happened between genes you're looking at. So that's why there's a focus on looking at the P0 gametes, and we have to figure out from the F2 genotypes, this blue bubble here on the right, we have to figure out from their genotypes what the F1 gametes were. Well, so the ratio of offspring, phenotypes or something like that, well, that's exactly what the genetic mapping information is. That's the genetic distance is derived from the ratio of these numbers. 128 to 247 is 67 to 58, for example. If it's 50 centimorgans, there are going to be an equal number of parental. So the sum of those two would be equal to the number of recombinants. So if we were assigning variables to those, you'd say that was X and that was X. The parental types will equal the recombinant types at 50 centimorgans. So that doesn't give us any information about whether or not the genes are on the same chromosome or not. We'd actually have to, punchline, you'd actually have to look at the chromosomes, maybe do some PCR or something like that to ask, are the two genes on the same physical piece of DNA or not? It's not information you can get out of genetic mapping. I'll take you and then have a question over here.
Could you also do something like if non-disjunction occurred, and so if you had B and L genes, they should always occur, but if you have B occur by itself and L didn't separate from a sister chromatid in one of the cells, that would be a strong indication that it's most likely on a different chromosome?
Sure. Yeah, I like that. So that's something I wouldn't have thought of, but seems reasonable to me. So you could look, although non-disjunction is relatively rare, so you'd have to look at, depending on the species you were looking at, hundreds or thousands or tens of thousands of offspring to get that sort of information. But yes, absolutely. So you could look at rates of non-disjunction to ask, are those two genes always traveling together or not?
Would there be a way to use the chi-squared? Because you're saying it should be half and half, but we always it doesn't always work that way, even if it is 50%. Would you want to use the chi-squared to make sure that's correct?
In either case, regardless of whether or not genes are on the same or different chromosomes, you'd still want to use the chi-square test. So again, yeah, the result of a chi-square test wouldn't give you the answer that we're looking for, unfortunately. Yeah.
I know that we saw in one of the classes that if we had a significant
Right. So when you see more parental types than recombinant types, that means they're linked. So this is the case where what happens when the number of parental types equals the number of recombinant types. That's when we get that calculation of centimorgans, right? So when the number of recombinants divided by the number of recombinants plus the number of parentals equals 0.5, 50 centimorgans. Just call that R. What does that mean about the values of R and P? If R divided by R plus P is 0.5, then R has to equal P, I think. I'm not a mathematician. So this is the problem, is that, and this is a quick trick for you, by the way. So if you look at a set of data, has anybody calculated the answer to this? I've suggested it as practice material for a couple of classes now, at least one. So who's got an answer? Genetic distance between A and B in this case. Nobody? Yeah.
I got 26.
Good. 26 centimorgans. So the recombinant types are the top and the bottom with parentals in the middle. So if you sum 7 and 6, the recombinant types, and divide by the total, which is 50, it's 13 out of 50 or 26 centimorgans. Okay. So A to B distance, 26 centimorgans. Yes, sir.
I'm slightly confused right now. Why is, so on this one, the 18,
Yep.
why is that the parental? Because it looks like the P not, but the one we just looked at, the parental was based off the F1 cross.
So it's always comparison of, what's the P0 gamete haplotypes? I haven't drawn them in, going quickly, to try to instigate you to ask me some of these questions, so I'm glad you did. So what are the P0 gamete haplotypes?
Capital A, lowercase b,
Capital A and lowercase b,
and then
little a, capital B. So those are the parental types. And we know that the F1 tester individual gives a little a and a little b to each of those F2s, right? So we cross out those haplotypes from all of the genotypes of the offspring, and what's left are the gametes that the F1 individual produced to fertilize to produce the F2. So now we know that this F1, if we wanted to write out their haplotypes, what their gametes genotypes look like, there would be 7 big A with big B, 18 big A with little b, 19 little a with big B, and 6 little a's with little b's. That's exactly what's left in that table right there. So which are the parental types? The big A with the little b and the little a with the big B. And the other two are the recombinant types. So in other words, let me put it one other way in case this helps. That P0 individual had on one piece of DNA, we assume, a big A with a little b. And that parent on the right had the opposite, a little a with a big B. And those chromosomes together went into that F1 individual, dihybrid. Capital A and little a, capital B and a little b, in that F1 individual, dihybrid. So now what we're doing by looking at these haplotypes down here is finding out what happened after recombination. So when recombination occurs during meiosis between those chromosomes, sometimes what are we going to get out? What haplotypes? Sometimes we'll get a big A with a big B. That's the recombinant type. It wasn't one of the original chromosomes, one of the original pieces of DNA that went into producing the F1 individual. There are new types of chromosomes that came out of the F1 individual's gonads to fertilize and produce F2s. So here's 26 centimorgans between A and B. So that suggests, it's less than 50 centimorgans, suggests they're on the same chromosome. Now what about this case? We're talking about the same gene A, now I've added gene C. Are they linked or are they unlinked? You don't even have to do any calculations necessarily. You should. I'd like you to. But just looking at the F2 data, the numbers of individuals that have 4 different genotypes. If these genes are unlinked, what's the expectation for the ratio of these 4 genotypes? It should be 1 to 1 to 1 to 1, given a dihybrid test cross. So if you want, you can draw out the Punnett square for the dihybrid test cross. It's a 1 to 1 to 1 to 1 ratio. So there should be equal numbers of those 4 if these genes are unlinked. Are they? No. So pretty sure they're linked. We should calculate a genetic distance that's less than 50 centimorgans. I'm expecting. So it's 20. So which are the recombinant types of the haplotypes down here?
Round, is it, are these round numbers?
Which numbers? The genetic distance numbers?
Yeah.
Yeah, that's fine. Yeah. This is not like chi-square test where you cannot round. So what are the parental haplotypes? Capital A with capital C and lowercase a with lowercase c. So down here when we look at the F2s, we can assign the capital A with the capital C, that's a parental type, and the lowercase a with the lowercase c is a parental type. And the other 2 are the recombinants. So you should get 12, 7 plus 5 in other words, divided by the total number of observations, which is 60? 12/60th, a fifth. So 20 centimorgans. Okay, so here's where I would definitely like you to do something. We just calculated the genetic distance between A and B. That was the previous slide. And now we've got A to C. So A to C is 20 organs. And A to B was, that's why I wrote this out in advance, 26 centimorgans. So draw me the genetic map that contains all 3 of those genes. What's the order from left to right? You can pick either, it doesn't matter which end you start on. So I heard, okay, so I've got one taker for ACB. So in that case, A to C is 20 centimorgans, A to B is 26 centimorgans. So what's the distance from C to B? Should be 6. But I'm going to put a question mark there because why? C could be on, in this case, changed colors, or could C be on the other side of A? Could it be CAB? C to A, 20 centimorgans, A to B, 26 centimorgans. So the only thing you should be looking for here is what? When you're trying to figure out, do you know the order of genes for sure? So these are the two different, with 3 loci, there will always be 2 options, 2 different orders of those 3 genes. How do you know that both of those maps are potentially right answers? They don't add up to more than 50. So 20 plus 26 is 46. If you got 2 genetic distances that were like 30 and 34, but in this case, either option is possible. It could be CAB from left to right. It could be ACB. For example, if I told you A to B was 30 centimorgans and A to C was 34 centimorgans, what would the genetic map look like? It can't be, A can't be in the middle, because then you'd have 34 plus 30 centimorgans, which is more than 50. So we could cross that out right now. So there's only one possible order, which would be ABC. In which case, the B to C genetic distance should be about 4 centimorgans. So in that case, you can tell without having to do the third cross. In this case, it would be the B to C cross, to actually experimentally figure out what's the distance between B and C. So here you go. Here's the third cross. Please do this on your own or collaborate with a friend, that's fine. Spend a few minutes. So there you go. So exactly the same cross structure, it's just the last 2 genes we need, B and C. Go ahead and work on this for a couple minutes and then we'll come back together and discuss. We'll figure out what the actual genetic map is. And do please try it, because if you have questions still, this is a good place to uncover those questions and to ask them. And of course, as always, there's an hour of office hours after class. So I'll help with the math a little bit. The total of observations is 200. It's too quiet in here, so either you're done or you've got questions. So let's move on. Which ones are the recombinant types? The middle 2, right? Small numbers, if there are any, are usually recombinant types. So even if you don't, and if you do not understand how to figure out the parental and recombinant types, come talk to me. But even if you don't know how to do that, when you see 4 sets of genotypes and 2 of them are much smaller numbers, those are usually the recombinant types. Usually. So 12 recombinants out of 200 total, map distance is 6 centimorgans. So with the A to B distance being 26, and the A to C distance being 20, then it makes sense that the B to C distance be 6, and that sums up to that map length we calculated earlier between A and B, which was 26. So now you know which order the genes are in, genetic map with genes A, B, and C. Any final clarifications requested, concerns, gripes? Okay, yeah, gripes, of course, always. So here's a quick summary for you to look over at some point. It's fine, but I think we've already beat that point to death. So here's a question I thought might be useful to summarize all this information. We've already talked about a little bit of this in review at the start of class. So here's a genetic map. I'm giving you the genetic map. We've got 3 genes, Q, R, and T. What's the cross, based on the video you watched for today, what cross would we want to set up to figure out the genetic distance between all 3 pairs of genes? What does it look like if you actually write out the cross? So 3-point mapping, yeah.
Is it tri-hybrid?
It's a trihybrid, which would be,
Homozygous, heterozygous times homozygous for all of them?
right, heterozygous for each locus, crossed to the tester. So I can add as many loci as I want, and you could do a 4-point test cross if you wanted to. Figure out the genotypes of 4 genes all at once. So every additional gene you add, you just add another heterozygous locus on the left to make a tetrahybrid or a pentahybrid, whatever. We won't do that, by the way. And then what are the genetic distances between the genes on this map? Another good way to summarize. If you took a guess, Q to R and Q to T are both what? 50. They're unlinked. So the Q to R distance you expect to calculate 50 centimorgans. They're on separate chromosomes. And then R to T, less than 50. Somewhere between 0 and 50 centimorgans. What would this have to look like if you were going to say that R and T were 50 centimorgans apart? How would I change that drawing?
You would take the tops, anything extra.
Yeah, so I'd have to move R and T to the very ends of the chromosomes. And that would, then you might reasonably say those are certainly going to be 50 centimorgans apart. Okay, time for a bit of discussion between you and your closest friends in the class, colleagues, cohorts, your neighbor. Brainstorm quickly, 2 minutes. Answer both of these questions. Is recombination good or is it bad or is it both? And can you come up with a circumstance, hypothetical, doesn't have to be real, but can you come up with a way to explain your answer? Is recombination good, is it bad, or is it both? So go ahead and talk about that. We'll pull back together in about 2 minutes. more interesting way of putting the question. Okay, so is sex good? Is it beneficial? If you were going to design the organism, would you want it to be having sex or not?
Yes. Yes.
Okay, so here's some yeses. Please defend your answers. So why is sex beneficial? Or why is recombination beneficial? Let's see. So here, and we'll move our way back.
It depends on how good the organism is to begin with. Like if it's already the perfect organism, you don't want to leave a chance for mutations to get in there that are going to harm it. But if it's something that could use improvement, then sex is probably going to cause improvement.
Interesting point. Okay, so to summarize, that answers both of the questions. Sex is sometimes good, sex is sometimes bad. So moving farther to the back of the class. Yeah,
So sex would be good because you have offspring that differs in genetic diversity,
correct.
survive better in the environment. But,
Maybe. So yes.
well, in the other case, okay, an offspring that can't survive in the environment has like, can't adapt to the environment or can't be self-sufficient.
Okay.
It's a laggard, they can't survive.
Fair enough. Okay. And the third point?
Well, they're basically the same somehow, because it causes, like, if you cause mutations to happen, but the genetic variety will also make mutations harder to happen. I was thinking about hybrid dogs and mixed breed dogs, when they say that mixed breed dogs are way healthier because of the genetic variety that they have, so I was thinking in that case, like,
So something about inbreeding and whether or not inbreeding is beneficial or not.
But if there weren't for human interaction, there wouldn't be pure breed dogs. All dogs would just mate with each other.
That's right. We actually caused the inbreeding of the dogs to occur, so that's a little bit artificial, sir.
It does come up with a little bit of point that the others made that it does depend on the environment. I remember like running across an article on a certain kind of lizard that actually has a parthenogenesis. So essentially it's cloning itself. So it really just comes down to the environment. Is the environment forcing it to change, or is its state already good enough for it to survive in that environment?
Okay, so in sum, what's the answer?
Both.
Both. Sometimes recombination's good. We talked about this when we started talking about recombination, this section of the class. And generally everybody was saying, well, it's useful because you're creating new genotype combinations and that's creating diversity in your offspring. Diversity, ooh, I should probably turn off the recording at this point. Diversity is not always good. So one of the examples, a hypothetical example was, say your genotype is perfectly adapted to your environment. So that you are as healthy as you possibly can be, and your kids are as healthy as they can be in that environment. Why would you want to create diversity? Because you're super healthy right now. The only time then you'd want to create diversity is when your environment changes. Think global warming, for example. So if you're an organism that doesn't do sexual reproduction and your genotype is perfect for life at 30 degrees centigrade, what happens when global warming occurs? When you have kids, they're genetically identical to you, and their kids are genetically identical to you, and your great-grandkids are genetically identical to you. But if the environment's changing, you're not creating diversity that could be healthier in those changing environments. So that's where recombination's good. Meiosis, new combinations of alleles, can help you adapt to changing environments. On the flip side, the opposite is also true. So if your environment's not changing, why would you spend all that effort making offspring that have diverse genotypes, that most of whom may not be perfectly suited for your environment? So sometimes recombination's good, sometimes it's not useful.
I like to look at it in terms of sickle cell and malaria. So I mean, obviously there is that external factor like malaria that pushes for genetic change towards sickle cell. But the negative side is that sickle cell it can also kill you. So you die of malaria, you die of sickle cell, but if you're a carrier for sickle cell, you don't die of either.
And that's so sickle cell and malaria and the hemoglobin mutations are an interesting exception to the rule in this case. Yeah, absolutely. Do you have a question? No, okay. Other questions, comments? So what we're going to talk about, I'm not done yet, by the way. I know you'd be shocked if I ended 15 minutes early. This was just a little preview of what we'll get into next class, which is the concept of genetic hitchhiking. We're going to move on to one more topic that I want to introduce today. But this is about genetic hitchhiking, that recombination is not always useful. Sometimes it is, sometimes it isn't. I'm going to switch gears now, so prepare your brains. There's one other thing that geneticists use genetic mapping for. This is something that I like to call phenotype mapping. It's not just genetic mapping as in calculating genetic distances between genes. That might seem like a rather trivial exercise to you, despite the fact that I've now spent like 3 classes talking about calculating genetic distances between genes. The goal of that, again, is just to estimate whether or not genes are on the same chromosome or different chromosomes. Maybe to you that's boring. To me, that's kind of boring also. But it's important to have that skill. In this case, this is a bit more maybe relevant. I'm sure some of you can think up of reasons that this is relevant. Trying to figure out what genes control certain traits. Phenotype mapping. We want to know what genes control certain traits that we're studying. So here's an example. This is called a simple trait. The word simple here means one gene. So if I talk about a simple trait, I'm talking about a trait, a phenotype that's controlled by genetic variation of a single gene. If we're talking about a complex trait, which is the next thing we're going to talk about, that's a trait controlled by multiple genes. So what's an example, do you think, of a complex trait in humans? You've looked ahead in the slides, but it's absolutely right. Skin color, eye color, height. What would be true? Hair color. Yeah, absolutely. A lot of our phenotypes are controlled by multiple genes plus the environment. It's not just a single gene controls the trait. What would it be like, imagine, close your eyes, imagine what the world would be like if human height was controlled by a single gene. And there were 2 alleles. What would the human population look like? You'd be tall or you'd be short. Or maybe you'd be medium if there wasn't simple dominance. So if you're a heterozygote, maybe you're medium. Right? So there'd either be 2 or 3 heights of people. That's not what we see. We see that there are a continuous number of heights, which tells a geneticist one of 2 things. Either that trait is controlled by lots of genes, not just one, because if there was just one gene, it would be binary, or the environment also plays a role in height, which we certainly know to be true. So in this case, a simple trait. We've got one gene, the gene is called A, up here in the corner. And so there are 3 different possible genotypes. You're either homozygous big A, you're heterozygous, or you're homozygous little a. And you can see the cartooned skin color, hair color, whatever you want to call it, phenotypes. Those are the colors of the boxes. So you have 2 black alleles, then you have dark skin color. If you have 2 of the white alleles, each of those dots representing a little a. Then you have light skin, and the heterozygote has an intermediate color. So you see there's the 1 to 2 to 1 genotype and phenotype ratios you expect from a monohybrid cross. That's simple. That's why it's called simple mapping. You get a perfect correlation between the genotype and the phenotype. If you have, that's what we're looking for, by the way, in the upcoming slides. One genotype corresponds exactly to one phenotype. So you have 1 black allele of this gene, you've got an intermediate skin color. If you've got 2 of the black circles, 2 capital A's in this case, you get the darkest skin color, hair color, whatever you want to talk about. So our goal is identifying the gene. So this is a case where we know the answer. This is what you expect when that gene controls that trait. This is what you would experience if you were actually doing this sort of a study in an organism. So here I'm diagramming the phenotypes of 8 different individuals. Except I'm not telling you which of the genes controls this skin color trait or hair color or whatever. So for 2 minutes till 11:45, figure out which of those genes, B, F, D, C, or E, controls this trait. This is a trait that's where the phenotype is controlled by one of those genes. So which of those 5 genes controls that trait? Go ahead, please talk to each other because this is difficult to work out, but need some feedback in about 2 minutes. This is not supposed to be easy to do. You don't know how to do this yet, but you might be able to figure out how intuitively. Yeah. So, is it E?
No.
Well, I think it's E because the individuals that have dark phenotypes each have an allele 1 of gene E. And each of the individuals that have light phenotypes have at least one E2, the second allele. So why is that wrong?
Those two would have to be homozygous at each one.
If there's what if there's what if there's dominance? So yeah, what evidence do you have?
Looking at the first individual and last individual, they both have the same combination.
Yes, so that's one way that you rule out that E is associated with that genotype, gene E is associated with that trait. Because if you get the same genotype in 2 different phenotypes, can't be that gene. Okay, what else? What's the answer? F. Okay. So in F, what did you notice?
The 2 with dark skin have the F1, and then the light skin have the F2, F2.
And that's what I'm talking about when I'm talking about just looking for the perfect correlation between the genotype and the phenotype. Every time you see one phenotype, it has one genotype. So which one's dominant?
Based on what we did earlier, the F1.
Well, it's actually, it's what? It's codominant, which is not something we've spent a lot of time, if any, talking about. But the heterozygotes have an intermediate phenotype. So there is no simple dominance happening here. This is codominance. Okay. So fantastic job. That's exactly how phenotype mapping works. You look at the phenotypes, and then you figure, this is how we know, for example, that gene F controls this trait. And then we've got the example of complex traits, where when you see not just binary differences or 3 different phenotypes, dark, medium, light skin color, or purple flowers, red flowers, but you have a continuously varying trait like height. So I don't remember when this picture was taken, but it looks like it was probably a long time ago, given what they're wearing. where the students in this college lined up based on their height, and so in 1-inch intervals, you can see how many students in this class were the different heights. This is what geneticists expect of these so-called complex traits, when it's not a binary phenotype. And that means that instead of asking scientists to do this phenotype mapping, look for associations between genotype and phenotype, which is easy when it's a simple trait, just one gene, it gets much harder the more genes you add. So you can see here, here's an example where this trait presumably is just controlled by 3 genes. And yet you have 7 different phenotypes. So if you have 1 gene controlling the trait, you can have 3 phenotypes. If you have 3 genes, you can have 7 genotypes. The more genes that are involved underlying this phenotype, the much more complicated the analysis gets. That's why we use computers to do these sorts of analyses and not just doing it by eye like we did on the previous slide. Finally, we've got complex trait mapping. Let me introduce this and then I'll take your question, certainly. Complex trait mapping, and this is what I'd like you to look at for next class. This is a diagram of 2 different chromosomes. So this parent is genetically homogeneous. That's why the 2 chromosomes is homozygous. The 2 chromosomes are the same color. So think of the colors as alleles. We've got a parent whose leaves have very few hairs or trichomes. We've got one other genotype, the homozygous blue genotype, where the leaves are very hairy. And you can see that their F1 heterozygote is in intermediate phenotypes. This is another example of codominance. And these scientists then, this is just a cartoon of real data, made a variety of siblings that have different fragments of the different chromosomes. So every time you see a single chromosome here, you should imagine that the other chromosome, these are all diploids, looks exactly the same. So each of these individuals has an interesting pattern of different blocks of the alleles, but they're all homozygous at every locus. So you need to figure out, your job is, what part of that chromosome from top to bottom, which allele, red or blue, if you drew one line across here representing a gene, which part of the chromosome controls the hairiness of the leaf? That's the job. Which genotype controls that phenotype? And then the other question is, try this. One more practice question on mapping. Okay.
On the color problem,
Yes.
you'll notice like 33 in a row with the same color. Is that assuming A, B, and C all have the same relative pigmentation of that?
Correct. See you all next time for in office hours.